So hello everyone, thank you for having me here. I will be presenting our work on Semantic Beacons, a framework to support federated querying over genomic variants and public knowledge graphs. This work was conducted at l'Institut du Thorax in collaboration with the LS2N lab. Both are located in Nantes. So first of all, I would like to give some background because my title is quite long. So we at l'Institut du Thorax work in close relationship with the hospital in Nantes, which is obviously an isolated health data silo. And our motivation for sharing the data inside is obvious because we would like to at least signal the existence of cohorts because they are relevant for rare or multifactorial disease research. And I will surprise no one by saying that sharing this kind of data is very challenging because data ownership, privacy, and just the inherently heterogeneous nature of health data. So we at Institut du Thorax have this cohort, which is called the ICON cohort, which motivated us to build the beacon, the semantic beacons. And inside of this cohort, we have patients with intracranial aneurysms. These are deformities of brain arteries. So the artery gets deformed and it is at risk of rupture, causing strokes. So these patients inside of the ICON cohort benefited from exome sequencing, a brain imaging exam, and have extensive clinical data description. And our challenge was to find ways, solutions, and standards to increase the fairness of this data, which was scattered and heterogeneous. This was one of the goals of the more broad project called Neurovask, and you can go to this website to find more about the overall project. So fortunately for us, we are not the only ones trying to share this kind of data in a fair way. So there were already several solutions existing. One of them, which we really liked and wanted to test, was the Beacon, which is a standard for genomic discovery and federated querying, which is promoted as a standard for, genomic discovery and federated querying, promoted by several organizations such as GA4GH and Elixir. And we really liked this solution because it provided, a standard, it was mindful of data ownership and privacy, and it also provided a model which helped us really structure our data and also provided a framework that guided us towards questions that we could or could not ask our data set. So on the other hand, we were also interested in using semantic web technologies. We wanted to be able to look inside of our health data silo in like more semantic way and transform it maybe into a linked data source, because we were especially interested in using already existing resources such as Wikidata, which contained gene location that we were interested in, and also we wanted to use UniProt KB that had protein function, and that is one aspect that we wanted to explore. So so now we thought about that data integration strategy, because inside of our ICON cohort, we had genomic variants, and we wanted to find out in what genes they were found and what was the proteins that they were coding for and what were the functions of these proteins. So what was our data integration strategy? Before telling you about our strategy, I have to tell you that integration of genomic data with knowledge bases happens all the time for people that actually do bioanalysis. So bioinformaticians, they routinely go look for, data to annotate their biological data on site. In the case of genomic variants, which we were especially interested in for the ICON cohort, PhDs and postdocs in our lab, they have the variant catalog and they go look into databases and look for frequencies in known population pathogenesis scores. location annotation, maybe the variant that was located in a patient that carries an aneurysm is found in a gene that has a certain interesting annotation, part of the overall interesting gene function. So what happens is that bioinformaticians and myself including, we actually handpick, copy, and import all this knowledge inside of our health data silo, and then we conduct our analysis and test our hypotheses. So our motivation for the semantic beacons was to try to do an easier integration of genomic data with knowledge bases, because we did not want to download a database or run an annotator. It involved massive data transfer and costly annotation process. This arrow here is on purpose messy, because it's sometimes a very messy process dependent on like annotated versions, database versions, and also just the parameters you put in and how you copy the data. So what we wanted was more on-the-fly retrieval of genomic variants within genes coding for proteins involved in functions that we were interested in. So our genomic variants were found in the data silo, and all the annotation that we were interested in are found in public knowledge bases that could already be exploited. So we thought about a use case, a simple question that would motivate our technical solution was to retrieve the variants found inside of our data silos that were annotated as found within genes coding for proteins involved in blood vessel formation, which was a function which was very relevant for people with brain artery deformities. Another word for blood vessel formation is angiogenesis, and I'm going to be using angiogenesis from now on. This was really our motivating use case. We have a data scientist that wants to ask the question, which genomic variants resides in genes coding for proteins involved in angiogenesis? And the data will come from several points, so let me walk you through each data source. First of all, the data scientists, we retrieve proteins with annotation with angiogenesis, and this can be done using protein UniProt knowledge base. Following this retrieval of the list of proteins involved in angiogenesis, we then go look in Wikidata to find the genes that code for these proteins, and we also want to extract the gene location. And all this is already is public, available, accessible data. And then we go towards the red part, towards our genomic variants, and then we want to query or to retrieve only the genomic variants that are found within genes that we previously retrieved. And we organized our genomic variants inside of the Beacon API because it was a standard and there was already a reference implementation and we could already use it. So we were really happy because the green part was already possible with existing solutions, as we said. As I said, so we went and looked at example queries in UniProt and found a couple of ones that we could copy. So one of them provided us with a way to query all the proteins involved in certain function. So here we used a gene ontology, which is a widely popular ontology to define, among other things, functions of proteins. And for angiogenesis, the gene ontology code is 0001525. So, and then we found another example query that made it possible to retrieve gene location from Wikidata using the protein ID extracted from UniProt KB. So we just wrote our own Sparkle query, kind of a mix of the previously two example queries where we answered all the green portion of our use case question. Just to give you a quick walk through the Sparkle query, line 16, I retrieve all the proteins. 17, I want only proteins that are found in human. And then I retrieve proteins that are tagged with the geoterm angiogenesis line 22. And then I bridge that towards Wikidata and retrieve the gene coordinate, which are start and chromosome, and also the genome assembly we were specifically interested in. So now, so this is just the result so that you can get on UniProt running that query. So now we had to solve the red portion of the question. So we used the beacon reference implementation of the API. We were very happy that it was already existing and there was an available implementation. We did no variant annotation inside when putting our data inside of the beacon. So our variants only had positional and allelic information inside of the beacon. And then we thought, okay, how can we make it queryable by Sparkle, and how can we integrate it as linked data? So how did we achieve that? Integrating beacon APIs as linked data. So we thought about an architecture of semantic beacons. So semantic, this is all the Sparkle portion beacons, all the beacon API. The green part already possible, as I showed you, and the blue part is, well, the beacon API, which was already existing, that holds our data from our ICANN cohort, that structures our data and is a standard framework. So we were really happy about that. So we had to find a plugin between the two. And to achieve that, we first wrote a semantic representation of the variants, then did an RML mapping for the beacon API, and did the actual integration of the non-RDF data found inside the beacon API. So first of all, the semantic representation of genomic variants, we thought about, well, data schema. Disclaimer, it was quite updated since submitting the paper. So we used popular expert domain ontologies such as sequence ontology and genotype ontology to actually represent the variant and its alleles. We also used Faldo to precisely represent the position of the variant, and we used SIO to represent some of the properties of this semantic representation. So once we had our semantic representation, we went towards RML mapping for the beacon API. So we just wrote a set of rules that define how to convert the JSON response from the beacon API towards linked data in turtle format that follows our semantic representation. And this is just a short snippet of the RML mapping. So if I ask our beacon API a question, do you have like variants surrounding this position? Maybe it will tell me, yes, I found a variant and its start value is this value in green. So then my RML mapping sets the rule that this green portion is an integer, and it's actually linked towards, with a Faldo position attribute towards something that is a Faldo exact position. And we did this for the start of the variant, the end of the variant, the alleles, and also the variant itself, obviously. And finally, once we had this semantic representation and RML mapping, we did the actual on-the-fly conversion between the beacon API and the linked data, thanks to an ODM TPF server. So what happens is that the TPF server converts triple pattern queries into HTTP requests that are compatible with our beacon API. And then the beacon API sends back responses, and these responses are mapped to RDF triples, again following our mapping. Then it is sent back to the TPF client that sends the response. So just a quick sum up of our contributions. Well, we thought about the data model to represent semantically genomic variants. We did some choice of domain expert ontologies. and made on-the-fly biological annotation of beacons or beacon API response possible. We were quite happy to be able to annotate genomic variants with gene ontology terms without any kind of data copying. So we also did the actual implementation of our use case. So if you, okay, up. So if you want to flash this QR code, you don't have to do it now. The presentation, I think, is going to be available, but there's a small demo server that runs a couple of queries, and among them is just the Sparkle query that is a translation of our motivation question. So everything from line 1 to 23 is just the green portion that was already possible thanks to existing solutions. So just retrieving the gene coordinates of genes coding for proteins involved in angiogenesis. And then we additionally retrieve here in red the genomic variants found inside of our beacon API. We retrieve the start and the end position and just apply a filter so that we retain only the variants found within genes that code for proteins involved in angiogenesis. And that way, we answer our motivation question. This is the result. Indeed, you only found variants found inside of gene coding for a protein involved in angiogenesis. You can run this query yourself if you'd like. And for the next steps, we would like to increase the speed of the query execution, maybe integrate new knowledge bases to add additional annotation. And we would also really like to add provenance metadata. I hope that I convinced you that we managed to do fresh on-the-fly biological annotation of genomic data, that way we minimize server-side costs by using externally maintained biological annotation. So, and that we actually did achieve the initial goal of increasing the fairness of our health data silo using community-agreed ontologies and standards such as the Beacon API. I would like to thank everyone for your attention, thank my colleagues for making this work possible, and if you have any questions, you're welcome.
Thanks, yeah, that was a great talk. I have a question, how versatile is Beacon? Can it be used for other kinds of data like proteomics data, spatial omics data, for serving that up on the fly as well?
I think there's a presentation coming up next about beacons, so that's why I did not tell a lot about them, but so far it can, correct me if I'm wrong, but so far it can only hold genomic data, so variant catalogs, but there's also, so we actually added semantics to only one of the 7 endpoints that the Beacon API provides. So that is the genomic position, but it also has API endpoints about metadata of the cohorts, about well, health-related terms, like you can add ontologies such as NCIT terms and also HPO maybe, and also tell what kind of tissues was sequenced. But as far as I know, there is no spatial, it cannot hold spatial genome RNA-seq data, if that's your question.
Thank you for the talk. Just a curiosity, you talked about the Nantes hospital. So you are fetching data only from the Nantes hospitals, or is there like a cohort of different hospitals that where you like integrate data? And
so the work that we did is part of the more larger project called Neurovask. So inside of this project, data was made available to us from this ICON cohort, and that's why we had access to genomic information and clinical data. And this cohort is actually from several hospitals, but it is monitored like by the Nantes hospital. So we do not have, let's say, fresh data coming in. We work we actually work towards kind of like a technological demonstrator to show that it's possible, and maybe if we manage to integrate it with directly with a plugin towards the hospital, it could be deployed. But for now, it's more like a technical demonstration that it's possible.
Hi, very cool work. I have just a very short question. For your data model about variants, did you use or based on some standard?
Well,
apart from the existing ontologies that you're using, the expert ontologies.
Why did I use the ontologies that I used? Or the schema? No, actually, there was
something else.
The actual data organization. We came up with this ourselves, and it was, at first it sounded quite straightforward. Okay, we just take the VCF and we transform it into a schema, but we kept updating and adding new details. So at some point we had to say, okay, this is where we stop and we don't go further. But the ontology, we tried not to add any new ontology, to use only what was existed. But the schema and the organization, that's our work.